Hey students, welcome you all on the behalf of Rahul Anand tutorial. So as you know that I am Rahul Anand and this is somewhat on my channel which name is actually the Rahul Anand tutorial. So now let's begin with the very interesting topics which are often being asked in terms of your Neet. Often the Aims, often the All India Pre-Medical Exam is the Human Genome Project. So these are the complex project as well as these do what? So these help in mapping the human genome at increasing fine levels as well as what? As well as preserving the information in terms for the databases and analyzing it in the future. So now let's understand about these means what are actually the roles of the Human Genome Project. So by understanding about these means the genetic engineering. So these are playing important role to understand what? To understand the here mean the DNA specific sequences in terms of the human body. So when we are talking about the DNA sequences, you have to understand primarily what are actually these means the genetic engineering. So what are genetic engineering? The very important process of what? The so the mixing of the here mean the ideal genetic material as well as removal of what? As well as removal of the non-ideal gene as well as the genetic material. So you could often understand that we are having the certain types of the features in terms of the ideals as well as the non-ideals, such as we are having the burntness like characteristics. Often in term we are having the royal color like characteristics. So these are playing important role in terms of what? In terms of the plastic surgery, such as we are having the different types of the hormones playing important role for maintaining the complexity of the white color. So these are actually what? So these are the thyroid etc. So we'll be understanding about this in the next. So what are actually the genetic engineering? The mixed management of the here mean the ideal genetic material as well as removal or either the abstraction of the non-ideal genetic material coming under what case? So it will be coming under the here mean the genetic engineering case. Often these are having literally the different mechanism in it. So let's try to understand what are actually these. So we'll be having what things? So we'll be having these means the DNA ligase as well as the polymerase chain reaction etc. Often we will be having the here mean the restriction agent. So in the case of the genetic engineering, we will be having what? So we will be having the restriction enzymes as like role of what? Playing important role as like the DNA seizures. After that you are having what? After that in the second we are having the vectors playing important role for the transportation mechanism. In the third we are having the ligase enzymes directly proportional for what role? Directly proportional for the role of the here mean the attachment of the ideal piece of genetic material. After that, in the fourth, you are having the PCR, mean the polymerase chain reaction, directly proportional for what? Directly proportional for the multiplication cases as well as the replication cases. So these are actually coming under what case? So these are coming under these means the genetic engineering case. So you could often popularly find here on the board here. By the way, let's understand about what are actually the deoxyribose nucleic. So in this you will be having the two of the polypeptide chain. As like in the case of the RNA, these polypeptide chains are quite one in nature. Often having the nitrogenous base in terms of what? In terms of the thymine is literally being replaced by the uracil. In what? In the RNA, in the ribose nucleic acid. Often these are having the angle of the bond of around how much? The 34 angstrom. As well as these will be having the nitrogenous bases such as what? Such as the adenine, the guanine, the cytosine as well as the here mean the thymine. So when we are talking about this means the DNA, so it will be having what? So it will be having the specific protein in it as well as in this case what happens? So in this case when the in term of the DNA sequences we will be having what? We will be having the overall genetic makeup of what? Of our organism as well as when what happens? When there will be the change in term of the genetic sequence, literally what will happen? The protein synthesis will be occurring. So you know about the protein synthesis very well, nothing to make you explain about that. And after that what will be happening? The overall characteristics with reference to our organism will literally be changed. So this is somewhat the condition why the organisms are literally being similar as like their parents. So you could often tell that we are having the what? The ancestor as well as the parents which resemble what? Which resemble the similar characteristics in terms of the species as like the morphological state as well as the anatomical state. So the parents will be resembling the similar what? Morphological and anatomical state as in like their here mean the offspring as well as the vice versa case. As well as when we are talking about these means in terms of our human being often So we are actually 99% literally similar to what? To our here, mean the parents. But they are generally among 1.5 to 2.4% of the variation. Literally due to that only we are quite a bit different in terms from our parents. Fine, so we'll be having the here, mean the brown eyes as well as the black eyes. Often you could have the curly hair, often the rowan odema, mean the skin color. So these are literally being caused by what? So these are literally being caused by these things only. By the way, now let's understand about these, means what are actually the ambitions of the Human Genome Project. So these literally played an important role to understand what? To understand the 3 billion pairs of the gene which are literally being present in terms of what? In terms of the human DNA, mean the human deoxyribose nucleic acid. Often by the mechanism of understanding the human DNA as well as the genes as well as the variations, we could do what? We could literally resolve the problems which are particularly being produced by the help of the non-ideal genetic material as well as plant the what? The ideal piece of genetic material to the ideal location. For what? For promoting the good quality in terms of the human health. Often by these mechanisms, what could be promoted? The life-extending mechanism with reference to the human development as well as the mechanism in terms of the survival. Best survival mechanism could be promoted as well as employed. So you could often find here on the board here. So this is the somewhat condition with reference when we are talking about these, means what are actually the Human Genome Project as well as what are actually the genetic material, often what are the DNA sequences. So this will be having the specific type of the protein on which the genetic materials will be present as well as these DNA pieces are literally composed of what? The 2 types of the polynucleotide chain. Often will be having what? The nitrogenous base such as the adenine, guanine, cytosine as well as the thymine. So I thought you have got the concept what are actually these. So if you like the work with reference to the education, often the lectures, so you could often subscribe, like as well as share as well as comment below in the text. Thus I will be knowing that what concept actually you are hungry for or what haven't you understood or either got. So very thank you with reference to what? With reference to the Rahul Anand tutorial. So you could often like as well as write these. So very thank you.